First, great things have small beginnings. In 1980, two men went for treatment at a genito-urinary clinic in West London. Both had urethritis. Nothing too unusual there, except the analysis revealed that among the microorganisms causing the infection was a tiny and previously unidentified parasitic bacterium. One so simple its genome was smaller than that of any other known organism. Such an elementary living entity immediately attracted enormous interest. Simply because it's so simple, Mycoplasma genitalium became one of the first bacterial genomes to be sequenced, and the first to have its DNA synthesized from scratch. And now it's become the first to be completely computer simulated. A team at Stanford have just announced they've modelled the cell and its entire life cycle. But besides leading to headlines like "Playing God, scientists simulate first ever 100% realistic sex disease bacteria," which was in, yes, that's right, the Mail, what do we gain by so laboriously mimicking nature? I spoke earlier to lead researcher Markus Covert from Stanford University's Department of Bioengineering.
The big thing to realize, I guess, is that for the past 60 years, let's say, ever since Watson and Crick and the double helix, we've basically been operating on an assumption that if we could take the genes, take the DNA, look at all of the genes, the genetic material, and isolate genes and determine what each gene is responsible for, what protein it's encoding, for example, what the function of that protein would be. If we could take all of that apart, there was this assumption that we could put it back together and understand cells, understand really life. And so we've been doing this for now many years, and there have been incredible discoveries and incredible benefits to humankind and human health in particular. But now we've kind of reached this point. Many people read or hear about a new cancer gene being discovered or a new Alzheimer's gene. And you hear about this all the time, and you might wonder, well, with all these discoveries, where are the cures to those complex diseases? And the answer is that cancer is not a one-gene problem. It is thousands of factors interacting in very complicated ways. And for us to understand a disease like that, we really need to start going back and trying to see if we can understand the whole cell in health and in disease. And so what my lab has done is taken all of the information that we could possibly find about each of the genes individually. We went through, looked at thousands of papers, tried to extract the most relevant data, and then put all of that information together into one combined model so that we could start seeing if that model would make predictions about the entire cell.
I can see why that's absolutely a desirable thing to do. I just thought that it sounded way beyond us because there are just so many different processes happening all at once, even in something as simple as this bacterium. How do you even know the scientific literature is across everything that's going on, and how do you get all of those different processes to feed off each other when you simulate it?
Well, to tell you the truth, even 5 years ago, I would have said that this was 20 years at least in the horizon. And so I'm just thrilled to see what the lab has been able to do. And it's absolutely right that our understanding of Mycoplasma genitalium is incomplete. And in fact, this is one of the things that we found in our study, that the model can be used to actually help fill in those gaps. So we can go through and make predictions, and in some cases they'll be consistent with what we measure, and that turns out to be most of the time, fortunately. But in the times when it's inconsistent, where we see a discrepancy, this can actually be like a red flag telling us this is a place where you need to have particular emphasis, where more experiments are needed. And so that was a very nice thing for us to see, that the model could actually accelerate the discovery process.
So is this a bit like climate modelling? You only know if you've got it right by seeing how your model performs compared to reality and then adjusting it?
Yeah, that's a good comparison. So in any kind of modelling, I think, and this might sound funny to say, but in some ways I'm happier to see when the model gets wrong than when it gets it right, because when the model gets some experimental data right, that means essentially that we understand that part of the organism. But when we see something wrong, it's basically pointing us to those little nooks and crannies, those parts that we don't understand but that could really be critical. And really the only way to do something like that is through a model-driven approach like this.
And do you have to try and get these processes down to the simplest, codest form? Because that's probably the way they are in nature.
Yes, so In some cases, that's true. In some cases, you have these dramatically simple and elegant processes that just seem to make a lot of sense. In other cases, evolution is a wonderful tinkerer, and so sometimes you find these incredibly complicated processes, and those then take more time and care. And so one of the critical aspects of our approach has been to try and use multiple mathematical methods to build the model. So in the past, even myself, I tend to focus on one tool, my favorite tool in my toolkit. But for this model, we found that the processes themselves were so inherently different that we made this model is actually a composite of 30 models, each of which was made with different and unique mathematics, and then all of those were woven together into one integrated whole. And one of the critical problems there is that many of them, there's a ton of crosstalk between these modules. We isolated them for the purposes of modeling, but in fact, they're deeply interconnected. And in particular, you look at something like metabolism, which is producing all of the building blocks of the cell. Those building blocks are needed by every single other module. And so to try and represent that, I actually had a great fortune having grown up in the Bay Area. One of my good friends worked at Google building Gmail, and as he was starting up a new company, he decided that he would also be happy to come work for us for a few months, just taking a leave of absence. And so during that time, he really revolutionized how we look at the code. And one thing in particular that he did was to kind of push us more into an object-oriented framework where each small molecule, each protein, each part of the cell was its own object. Now that was particularly interesting because we found out later that the object-oriented approach developed by Alan Kay, who was building up one of the first object-oriented programming languages, he said that he actually had a biological paradigm in mind when he developed that coding framework. And so it was really fascinating to me to see, wow, biology inspired his approach to making this object-oriented programming language, and then in turn, the object-oriented programming approach enabled us in many ways to be able to integrate and produce this entire cell. So it's all kind of come full circle, as it were.
And are you now confident you've got it right?
No, in fact, I'm sure I haven't got it right. So I was very gratified with how much we're able to predict with confidence. And even the new experimental data we're currently generating in the lab has been very supportive of the predictions that we've made. But kind of like the Human Genome Project itself and all of the genome projects, when the genome is first described, it's very explicitly called a first draft. And this model should also be thought of as a first draft. We want other people to look at it and criticize it. In fact, for those of you out there who like to hack, it's completely open source. You can download it from our website. We're happy to help anyone get started. In fact, there's a Hacker News blog where I've been eagerly watching the posts as people have been taking this model and downloading it and actually running it, which has been tremendous. I strongly hope that people will, from the computer side, they'll look at trying to speed things up, and I'd be thrilled to see that. And on the biology side, I'd be absolutely thrilled for people to come in with their own models.
And what do we hope to gain from this in the long term, apart from it being a stepping stone to more complicated life forms?
I think there are two things that really have dramatic potential, and one of them is just the ability of modeling to dramatically accelerate biological discovery. Okay, like I told you, we've done a lot of things at the single gene level, but if we really want to take discovery to the next level, we're going to need to consider systems, biological networks, biological pathways, biological systems. And to do that, we need a computational framework. So that's one. And two is just the idea, now that we can synthesize a chromosome of a cell, the idea of using these kinds of tools to eventually start designing bacteria. Every other engineering discipline has computer-aided design tools to help make sure that the design of new products is safe and doesn't have unwanted side effects, and really just to make sure that you can make the best possible design. And we're just in the very infancy of being able to do that, but I think the advent of a model like this and these new synthesis tools makes something really something that you can now imagine in the near future.
Markus Covert stimulating our imagination through simulation. And there's links to more on life in silico via the Material World web pages within bbc.co.uk/Radio 4.